Hi everyone, my name is Wei-Lee Wu. I'm a third-year PhD student at Purdue University studying chemical engineering. I work in Dr. Zoltan Naj's Crystallization Process and Control Research Lab. Today I'm here to tell you a short story about the utilization of quality by control method with easy viewer image analysis for rapid process design of an agrochemical crystallization process. This work was done in collaboration and sponsorship with Corteva Agriscience. Crystallization is a common method of purification and particle design step in agrochemical industry. One of the major challenges experienced are long cycle times in crystallization, filtration, and drying. Long filtration times are directly impacted by poor control of particle size and shape. Poor impurity rejection and solvent inclusion are also problems that occur during a crystallization process, which can lead to an increase in drying cycle time or even cause the product to be off specification. Traditional methodology to address these issues involve the quality by design methodology. While it works, QBD requires extensive design space testing, which requires large amount of material and time. With the help of process analytical technology tools and feedback control loops, we propose to apply quality by control methods as a faster methodology to find the optimal operating procedure. Through collaboration with Corteva Agriscience, our case study process is a batch anti-solvent and cooling crystallization process. The model compound, needle Z, is crystallized over 8 hours from a mixture of a solvent and an anti-solvent. In a typical crystallization run, very fine needles, as you can see from the microscopic images on the right, are generated, causing long filtration time of over a day in a typical pilot plant production. It should be noted that impurities are generated during the crystallization process. The motivation for this work is to design a model-free QBC methodology to design a crystallization process that can increase the particle size, which can reduce filtration time while minimizing impurity generation. The experimental setup of the crystallization process here at Purdue involves using multiple inline measurement tools to observe the process dynamics. The ATR UV Vis probe measures solute concentration. The FBRM, or the G400, was used to measure the particle counts and core length distribution. The PVM and the EasyViewer provides particle image shape and morphology analysis. These PAT tools are hooked up to a supervisor computer that communicates with all the hardware to provide feedback control to manipulate the process temperature. Offline tools are also vital in characterizing the crystal product. Off-site HPLC was used to monitor impurity levels in the crystal cake. The impurity seed generation from a typical production batch is around 0.05 weight fraction. The Malvern morphology G3 and its wet cell dispersion feature was used to characterize the particle size to generate number-based size distribution. With these tools, we observed a scaled-down original recipe experiment in a 500 ml crystallizer. The inline and offline imaging shows very fine needle particles generated with a mean size of about 34 microns. A simple Buchner funnel filtration took about 6.5 minutes to filter. The traditional methodology to tackle the small particle size issue is the model-free QBC direct nucleation control. As the crystal mean size is inversely proportional to particle counts, DNC utilizes preset limits of particle counts detected via FBRM to dissolve small particles and maximize growth of larger particles through oscillatory driving force trajectories. Typical DNC temperature trajectories would yield oscillatory behavior, as seen in the bottom left figure of particle counts. This is the black line versus time. One can see that temperature changes with the directions as the particle counts reach a maximum and minimum threshold. In this experiment, the mean particle counts are set at 35,000 with a 5,000 particle count boundary. However, there are times in which the process will not converge, leading to infinite cycles. The cause of infinite cycling can be explained by the rapid increase in aspect ratio of the needle particles by growth. The FBRM would detect such change as nucleation, which can prematurely trigger dissolution from the feedback control. The microscopic images of the particle show that the DNC method can grow the needles to a sizable length and width. However, high impurity C was detected in the HPLC after greater than 36 hours of thermal cycling. Thus, an upper bound of crystallization batch time is set. Concentration is important to understand the process dynamics well. Thus, a good UV to concentration calibration model is required to observe concentration changes. Previous research has demonstrated that DNC trajectories can provide useful information about the model's compound solubility, primary nucleation zone, secondary nucleation zone, as well as growth. Absorbance readings from the ATR UV probes are affected by the system temperature. One can see that increase in absorbance during cooling of a supersaturated solution. As well, on the figure on the right, to decouple the temperature effects of the probe, a bivariate polynomial model was used to fit the experimental data. The first derivative of the absorbance reading was used as it provides the best fit in the calibration model. This model is then used for future experimental design and QBC methods. As the morphology of needle Z is high aspect ratio, an image-based particle analysis approach was used. The original intention was to use a dry dispersion method for characterizing these particles. However, needle Z tends to agglomerate upon filtration. During dry dispersion, the particles either break due to high pressure or does not disperse well for image analysis. Thus, an alternative approach was used. The wet dispersion process starts with sampling in triplicates of the dried cake and dispersing it in water. The solubility of needle Z is negligible in the water at room temperature. Then the vials are put on a rocker overnight to allow for gentle dispersion. The next day, the vials solution are then injected into the morphology's wet cell accessory. The particles dispersed in the bottom right image are then measured by the camera to generate a number-based distribution. Here we have a good comparison of length versus circular equivalent diameter. The length distribution was chosen as a default particle size distribution based on the high aspect ratio nature of the particles. Another usable method of model-free QBC is direct nucleation control combined with supersaturation control, or known as SSC. SSC utilizes concentration feedback control to promote growth of particles by holding the supersaturation level constant while changing the cooling rate and the process system temperature. SSC requires a robust UV to concentration model to monitor the supersaturation. In the controller, the batch time of this process is set to 24 hours to minimize impurity C generation. The thermal cycling from DNC increases the aspect ratio and mean size of the particles. After DNC, as you see with the images of the blue star, you can see long needle-shaped particles. However, the slow cooling with SSC inevitably triggers secondary nucleation, which led to fine generation, as after SSC, you can see some of the original seed crystals, but a lot of the fines are generated. In the microscopic image, fine particles are attached to the large seed crystals from DNC, indicating that this system is a nucleation-dominated system. Thus, thermal cycling is the best methodology to tackle this problem. It's also worth noting that this experiment did improve the filtration and mean particle size from the original recipe. If textbook model-free QBC methods don't work on this compound, what if we force the system to converge and reverse engineer a model-free QBC method that will make it work? By implementing a converging temperature trajectory while observing the process dynamics with the PAT tools, the turbidity reading from the Easy Viewer image analysis provided a converging statistic. This is seen in the green color oscillating turbidity values that converge at around 0.8. The turbidity reading is generated by the PVM Easy Viewer image analysis package from IC Vision. As the number of crystals increase, the pixel intensity of the images become higher, which correlates to an increase in turbidity reading, vice versa for dissolution. This simple converging thermal cycling led to a large increase of particle size while keeping impurity C at a reasonable level. It is worth noting that the FBRM counts always reach the same plateau regardless of the growth of these crystals. We're hypothesizing that the FBRM has trouble scanning the length of the needle-shaped particles. There is a higher probability of the FBRM laser to detect the width of the crystals along the length side. As the crystals grow lengthwise and barely in a width direction, the FBRM can only pick up minor changes in its path length. This is also corroborated with the stagnant mean core length profile as seen in the red figure. Through several iterations and fine-tuning, I present to you turbidity direct nucleation control. Like DNC with particle count, the controller was given a set boundary for the turbidity values. These values were designed based on the converging temperature trajectory. During the iteration, it was found that a manual high temperature cycle in the initial cycle is necessary to dissolve the polarly mixed zone after nucleation. The TDNC approach generated crystals far larger than any converging methods done prior while keeping impurity at a controlled level. A simple Buchner funnel filtration presented double the filtration rate than the original recipe run. To test the robustness and usability of the new method, TD&C was tested on a range of solvent compositions with needle Z. On the solubility surface, which is measured gravimetrically, the concentration in needle Z changes with the ratio of solvent and antisolvent. These solvent compositions mirror the variations of feed composition that needle Z comes in from the previous unit operation. The original feed composition is 1:8. Overall, the TD&C method converged for all experiments with similar turbidity profile. In the process temperature figure, as seen on the right, as the antisolvent composition increases, the convergence time of the TD&C increase. It is also worth noting that the process system seems to have converged at an earlier point for the experiments with higher solvent composition. This is the 1:7 and 1:8, as it completes around the 15-hour mark. There was not a discernible difference between the mean size, filtration time, and impurity generation among these experiments. In our open loop and scale-up study, we took samples before and after the last cycle to examine differences in the particle size. This is denoted with the red star and blue star in the figure on the left. In the small-scale 500 milliliter open loop experiment, we see that the particle size is comparable before and after the final cycle. The particle size between the closed-loop TD&C approach and open-loop methods are similar. However, when scaling up, the mean particle size before the final cycle is smaller than after. This indicates that the scale-up of the needle Z process may not be a direct translation. Furthermore, differences in the turbidity between the 2-liter and 500 milliliter experiments also indicate that the process dynamics are different. These shortcomings in scale-up indicate future work with a model-based approach can be beneficial and may address the complexity behind scale-up in crystallization. The goal of this work was not to just increase the mean particle size. It's actually to demonstrate that this QBC method can decrease filtration time. With the use of AWL's pocket filter, we can standardize the filtration method across various experimental conditions. With the direct comparison of the original recipe, the open-loop small-scale experiment, and the open-loop large-scale experiment, we can see the filtration over time plots. On the bottom left, the massive filtrate over time indicates that the QBC approach substantially decreased the filtration time of the original recipe slurry, which reduced the moisture content of the cake and can also improve drying cycle time. Through a quick analysis of Routh's equation derived from Darcy's law, the average cake resistance derived from a linear regression is an order of magnitude smaller with TD&C approach compared to the original recipe. To briefly summarize my talk here today, our goal was to use QBC design to reduce process cycle time of an agrochemical crystallization process. Because various traditional methods couldn't yield desirable results, an image-based turbidity direct nucleation control method was developed. The robustness, reproducibility, and usability of the new method was tested by varying solvent compositions, performing standard open-loop and scale-up experiments. Finally, a standardized filtration study demonstrated reduction of filtration time by 2.5 times compared to the original recipe. For future work, we would like to propose a model-based QBC approach to optimize the crystallization process while investigating scale-up approaches with coupled filtration and drying. Thank you for listening to my talk today. I would like to acknowledge Corteva Agriscience for their support and collaboration opportunity. I would also like to thank my advisor, Dr. Naj, my collaboration team, as well as my research group, CRIPSIS, for their help and support. I would be happy to take any questions.